Dr. Eric Raabe. I work with Johns Hopkins. As you heard, it's really a chance to actually make a difference for patients by accelerating drug development. So that's really what it's about, is to try to get new therapies to patients faster. So if we can do that by having an intermediate step between what happens in the dish and what happens in the mouse, then we could potentially decrease the amount of time and money that it takes to do mouse experiments, use less mice, and then accomplish it all in a way that actually gives us more sort of richness of data, because we can actually watch, because the fish are clear, you can watch it all happen in real time. So you can watch the DIPG cancer cells move around or not. If your drug blocks migration, then they won't move around as much. And if your drug kills the cells and doesn't kill the fish, then the number of fluorescent cells or luminescent cells will decrease over the course of several days, which is what you want. You want to not so much stop it in its tracks, so that would be okay, but you really want to actually make it get smaller, because that's really what we want to do. We don't have that many patients to do clinical trials in, and that's a good thing, but you also realize that it can take years for a clinical trial to actually finish. So you would prefer to try to do clinical trials with drugs that actually give you the best chance of making a difference for the patients. The more that we can tie together the people who are working in DIPG, the better. A lot of us know each other already, but there are others who are new to the area who are coming, and that's really exciting to be able to see the progress that's been made. But I think that there's always room for more collaboration, and there's room for more ideas also. So one of the things that's pretty exciting is when you can take something from another field or bring an expert in another thing and bring it to the particular type of pediatric cancer that you're studying, because that, if you can find an application for whatever sort of widget can then be brought into it, so in this case it's like zebrafish biology, then you can really apply that to a problem that exists, which is the sort of bottleneck for new drug development and new drug sort of discovery and the iterative process for doing these kind of things. As Nick Gottardo said, like, it's really an ideal thing to try to do combination studies in the fish, because you can generate 100 fish in a day, and then that gives you a chance to have cohorts that might have 20 fish in each arm, which you could then find out the answer within 7 days, as opposed to having 40 mice, which each mouse is pretty expensive, and so the minimum cost is about $50. So you're really getting into the thousands of dollars very quickly for those kind of things. So I think a fish, because they can generate a ton of eggs really quickly, and then the next week you can mate them again and they can have more eggs, that makes it so that you could really, in a fairly inexpensive fashion, really move things forward. So this research wouldn't be happening without the Cure Starts Now, because they've provided this critical funding to move this project forward. And so we wouldn't have been able to develop the models that we have in zebrafish and to do some of the really interesting experiments that we've been able to accomplish without funding from the Cure Starts Now. And we're hoping that this project will continue, because we still have a lot that we need to learn. Further, studying new models, meaning more DIPG cell lines to be put into the zebrafish, and also being able to test how this works with the immune system, and coming up with a way of doing high throughput drug screening using the zebrafish as sort of an intermediate between what we can do in cells in culture and the actual mouse experiments.